Hello everyone. My slides are visible, and am I audible to all of you?
Yes, ma'am.
Okay. We have to now start the most important part. We have already uh, have studied about the role of microbiota in protecting our body against different infections. That how our body fight against different infection, different pathogens, the bacteria, fungi, or any other foreign material. So now we should know about the exact or the specific mechanism, that how our body achieve all those things. So in this today's lecture, we will study about the specific mechanisms, specific molecules that plays a vital role in protecting our body against different infection. So before that, we should solve some questions which is based on yesterday's topic, and this is very important and basic. So first question is, which of the following immune functions is directly influenced by gut microbiota? Production of RBC, that is red blood cells, regulation of T regulatory cells, increase in platelet count, or inhibition of melatonin release.
Regulation of T regulatory cells.
Yes. So right answer is regulation of T regulatory cells. So we have studied yesterday that the gut microbiota plays a vital role in the stimulation of T regulatory cells, which are involved in different inflammatory responses. If our body encounters any kind of foreign material, be it a pathogen or any other thing, so our body start producing or stimulating the T regulatory cell. As T cell is of three types: TC cell, T T helper cell, and T regulatory cell. So T cytotoxic cell, T helper cell, and T regulatory cell. So these T regulatory cells are involved in inflammatory responses, which our body provides whenever our body encounters a pathogen. So our gut microbiota directly will influence the regulation of the T regulatory cells. So gut microbiota produces metabolites like short chain fatty acids, especially butyrate. We have already studied this yesterday, which promotes the development and function of T regulatory cell that will function as a immune tolerance, and it will prevent the autoimmune diseases. When our body will consider our own cell as non-self, and they will treat it as a foreign material, so they will start reacting against it, and our body will start having inflammatory responses. So all these gut microbiota are involved in the regulation and stimulation of the T regulatory cells. Now second one, short chain fatty acid SCFAs produced by gut bacteria primarily affect the immune system by destroying T cells, enhancing IgE production, promoting anti-inflammatory responses, or inhibiting phagocytosis. What SCFAs do? How they influence our immune system? We have just now read here that our gut microbiota produce a metabolite like SCFAs, short chain fatty acid, for example, butyrate. So what butyrate will do? It will be directly involved in the inflammatory responses. It will stimulate the regulation of T regulatory cell, and T regulatory cell is involved in what? Inflammatory responses. So for this, SCFAs will do what? They will start promoting the anti-inflammatory responses. They will start producing certain metabolites which will reduce the responses of our body against the pathogen, because ultimately our body should be in a equilibrium. It should not be always in a mode of reactiveness or responsiveness, because whenever our body fights against a pathogen, it will drain all of the energy of our body. So our body will also having certain drawbacks in fighting against any of the pathogens. So our body needs to be in an equilibrium state. So whatever gut bacteria will do, it will promote anti-inflammatory responses to fight against the pathogen, to protect the body, to protect the body's homeostasis. So right answer is option C. SCFAs like butyrate, acetate, propionate promote anti-inflammatory effects by inducing T regulatory and suppressing the pro-inflammatory cytokines. It will reduce or inhibit the inflammatory cytokines like interleukin 6 and TNF alpha. So you have to remember that anything in our body which is involved in the inflammatory response, so our immunity will do what? Our immune system will stop that or inhibit that particular molecule which are involved in our inflammatory responses, because our body needs a equilibrium state to remain healthy. Now third question. So dysbiosis of gut microbiota has been associated with which of the following immune related diseases? Asthma, type 1 diabetes, inflammatory bowel disease, all of the above. So tell me the answer. Dysbiosis or the perturbation of the normal gut microflora will result in which kind of disease? All of the above, because asthma is also a infectious disease, type 1 diabetes, inflammatory bowel disease, any of the disease may occur, because whenever our immune system or our immunity will be decreased, it can result in any of the mentioned diseases. It can cause asthma, because whenever you will encounter some dust particle or dirt particle or any other foreign material, your body will start reacting towards it. And type 1 diabetes, your own cells are not producing enough insulin, or they are not even reacting in towards it. Type 2 diabetes and also IBD. So any of the disease is possible when you are having an low immunity. Now coming back to the topic, main topic, which we have to start today. So the topic is role of microbiota in immunity and inflammation. We have already studied so many things yesterday about how the microbiota protects us against different diseases, but in this lecture we will study about the exact mechanism. So you know about the gut homeostasis is maintained by the presence of obligate anaerobic bacteria, specifically Firmicutes and just a minute, I have to change the color. So our body, if our body has an intact gut microbiota, so our body will be having the Firmicutes and the Bifidobacteriaceae. Its concentration will be high, Firmicutes and Bifidobacteriaceae. But when there is an increase in facultative anaerobic like Enterobacteriaceae, which is a common marker of gut dysbiosis. I've already told you that you should observe the different medical reports of any person available in your home, because they told you about so many things in a medical perspective. Because like normal CBC test, it has so many parameters: RBC, WBC, WBC2 granulocyte, granulocyte. So you should what is the normal range of basophil, eosinophil, and what is the range of lymphocytes, neutrophil, everything. So hemoglobin range, normal hemoglobin range in a healthy individual. What is the range, exact range of platelets? So iron level. So all those things you should know about. The normal blood sugar report, the fasting blood glucose, the postprandial glucose, that is fetal glucose. So you should know about all those things, and it is all written in the report, like the observed patient sample and the normal reference or biological or the normal level. So through that report, you can be aware about what is the exact range in our body to be a normal individual, normal healthy individual. So through different reports, we learn so many things. So you should always take care of those things. Try to learn from different things available in your home. So if the concentration or the composition of Enterobacteriaceae will increase, then it is a common marker of gut dysbiosis. And if it is only Firmicutes and Bifidobacteriaceae, then it is a marker of gut is healthy. So you should know about the term facultative and obligate. So facultative and obligate means what? If I am saying obligate anaerobic, like it will not grow in the presence of even a little bit of oxygen will prevent or inhibit the growth of such Firmicutes and Bifidobacteriaceae. So obligate means only grow in the absence of oxygen. Obligate means they cannot do any flexibility kind of thing. No, if they want obligate aerobic condition, they require oxygen only. If they require obligate anaerobic condition, then a little oxygen will hinder their growth. Now facultative means they are flexible. Even if it is available, a little bit of oxygen will it is available, it will not hinder the growth of the bacteria like Enterobacteriaceae, because it is a facultative anaerobic. It can also go in the presence of little oxygen and also in the presence of no absence of oxygen. So under gut homeostatic condition, the IEC is intestinal epithelial cell. The cells of our normal gut normal. If your gut is healthy, if your gut is not kind of kind of disorder, damage, nothing, your gut is fine, then your intestinal epithelial cells will synthesize what? The PPAR gamma. PPAR gamma is what? Peroxisome proliferator activated receptor gamma. So your body will start synthesizing what? The short chain fatty acid is the butyrate. So this is a condition when your gut is healthy. So you should know about this thing. We are firstly talking about the gut homeostatic condition. It means if your body is having a normal gut microbiome composition, your body will start producing what? Start producing AR gamma. So this is a nuclear receptor that will act and it will play a and it will start the synthesis of the short chain fatty acid, that is butyrate. So this butyrate is nothing but a short chain fatty acid. So this PPAR gamma will help to maintain a local hypoxic environment by encouraging oxidative phosphorylation in the colonocytes, the cells of the colon, and beta oxidation of SCFAs by the mitochondria. So many things are happening here. What PPAR gamma will do? PPAR will do, it will promote the hypoxic environment, like the absence of oxygen. So it will encourage what? Oxidative phosphorylation and the beta oxidation of the short chain fatty acid by the mitochondria. Now this obligate anaerobic, it means it will not grow even if little bit of oxygen will be present. So it is obligate anaerobic. It does not require even oxygen, no, nothing. So SCFA producing bacteria grow vigorously in such an environment, while the facultative anaerobic enteric pathogen growth is suppressed. Now, if there is an obligate anaerobic condition, so bacteria will grow. Which bacteria will grow? Only the bacteria which are capable of synthesizing short chain fatty acid will grow, and the pathogenic bacteria that require oxygen, because it is facultative anaerobic. So if you are maintaining a condition that has no oxygen, suppose there are two chambers in which you have provided some amount of oxygen. This has little bit of oxygen and this has no oxygen, no O2. This has so many molecules of oxygen. So this will promote the growth of pathogens. Why? Because the pathogens are what? They are facultative anaerobic. It means they can also grow even if little amount of oxygen is present in the environment. But the obligate anaerobic, which are the, you should know about this bacteria, these are the good bacteria. These obligate anaerobes are the local or the good bacteria of your normal gut. If your gut is in homeostatic condition, so you need to maintain the anaerobic condition, obligate anaerobic, because your SCFA producing bacteria can only tolerate the absence of oxygen environment. If a little bit of oxygen will be present, then which type of bacteria will grow? Facultative anaerobic bacteria will grow, which will cause infection in your gut, and therefore will be initiated. So you should know about maintaining an obligate anaerobic condition. So SCFA producing bacteria will grow. It means good bacteria will grow and body. And if we are providing oxygen in the gut, so we are providing conditions for the pathogen to grow. Now, this PPAR gamma activation decreases the nitric oxide synthase levels in the intestinal epithelial cells, hindering the manufacture of both inducible anosynthase and nitrate, critical sources of energy for facultative anaerobic pathogens. So now you should know about this also, that PPAR gamma do what? First of all, it is encouraging the oxidative phosphorylation. Let me highlight the conditions. Blue color. So it will encourage the oxidative phosphorylation, the first thing, and the second thing, beta oxidation of fatty acids by the mitochondria to produce what? ATP. Now, what the third thing is happening here is PPAR activation decreases the nitric oxide synthase level. In the everything is happening inside what? The intestinal epithelial cells. So the intestinal epithelial cell will do what? The anosynthase will be hindered, the synthesis of the hindering of the manufacture of both inducible anosynthase and nitrate, which are the critical sources of energy for the facultative anaerobic pathogens. So now the cells or any kind of any cell require glucose to grow in the body. But some pathogenic microbes or some of the pathogen rely on different material, different groups like nitrate to grow in the body. So these nitrate will be splitted to more simple molecule and then they will be consumed by the facultative anaerobic pathogens, which are the main for our body. So PPAR gamma is doing what? It is not providing a condition to provide energy sources for the growth of facultative anaerobic pathogens. So I hope I am explaining it in a simple, proper way. I'm trying to explain is that this PPAR gamma is doing a very good thing. It is doing motivation, will not support the growth of facultative anaerobic pathogens. Because if oxygen is present, then we have studied here, if a little bit of oxygen is present, facultative anaerobic bacteria will be grow. It can grow. So if pathogen, so if its growth will be increased, then it will cause inflammatory responses and different infections. So our protagonist or our hero is obligate anaerobes. So obligate anaerobes concentration we have to increase. So how we can increase the concentration? We have to eliminate each and every oxygen molecule present over there. If we are allowing even a single molecule of oxygen, then it will promote the growth of these facultative anaerobic bacteria. Now, the third thing which is done by that it will increase the NOS2 levels in the intestinal epithelial cells, and this will do what? This will not provide any kind of energy, because every cell requires energy. Like we need energy to do any task, any such thing. So any bacteria, any cell will require energy to do its normal metabolic machinery. So if you are cutting off that particular energy source, then that bacteria will not be able to grow, not be able to do its metabolic thing. So it will ultimately be reduced in that particular gut area. So this is the adaptive mechanism adapted by the gut microbiota to prevent infections in your body, to prevent and boost your immunity. So these are the methods, these are the mechanism through which our microorganism available in your microbiota helps the body to fight against different infections. So if you are asked a question that how these things are going on, how if you are saying that the gut microbiota has a role in protecting or our body against different pathogen or boosting our immunity. So can you tell me any mechanism? So these are the mechanism. You can say that our intestinal epithelial cell is responsible for so many things. So what it is doing? Firstly, everything starts from the synthesis of short chain fatty acid. Like they are synthesizing, first step will be what? They are synthesizing short chain fatty acid. Short chain fatty acid is synthesized. Then it will synthesize the peroxisome proliferator activated receptor gamma, the PPAR gamma, which is very much important. So this PPAR gamma is a nuclear receptor that will act as a transcription factor. So it will play a key role in the regulation of the gene expression related to energy metabolism, cell differentiation, and also the inflammatory responses. So as you all know, this PPAR gamma has a role in so many different things. Like it can control the cell differentiation, it can control the fat accumulation in our cell, it can also control the inflammatory response. So this PPAR gamma is a very, very important transcription factor known in immunology, that it protects us from so many things. It has so many vital role in our body in the context of immunity also, and in the context of cell biology also. So it is involved in so many biological processes. Now this PPAR gamma has also a role in the diabetes kind of thing also. So if you are saying that your PPAR gamma level is not sufficient, or if you are saying that your PPAR gamma is not performing its function well, like it is impaired, that you can say that your gut homeostatic conditions are not maintained. So if you are a healthy individual, then your PPAR gamma will be in a good amount. So this PPAR gamma needs to be in a good condition to remain us in a healthy state. So in simple line, you can say that impaired PPAR gamma function can contribute to inflammatory diseases. Now यहाँ से क्या line आया main? That PPAR gamma is a critical regulator of our immune system, and you can say that if your PPAR gamma is impaired, I am saying it again and again so that you can write it in your notes. Impaired PPAR gamma. If any individual is having impaired PPAR gamma, then you can interpret it as that it has inflammatory responses in the body. Inflammatory disease will occur. And if you are having normal PPAR gamma, then no disease can occur in your body, because PPAR gamma is producing so many different kind of effects in your body. Like it is promoting the oxidative phosphorylation in the colonocyte to maintain a what? Hypoxic environment. Hypoxic environment means a low oxygen condition in the cells of our gut. and it is also promoting the beta oxidation of the short fatty acid by the mitochondria to produce the required energy which our body needs to do the normal metabolic machinery. We are doing a lot of things in our day-to-day life, so we need energy for each and every. We need ATP, the energy currency, for each and every task we are doing in the normal 24 hours. So for that, our PPAR is promoting the beta oxidation of the fatty acids. Now, third thing is this mechanism. PPAR gamma activates the decrease in the nitric oxide synthase level, and this is the critical regulator because this acts as a energy source for the facultative anaerobic pathogen. Now from here you can also correlate to more things that in our gut. Write it down. I don't have any space here, so let me clear first. All these points are very important because I have to explain the flow chart. So therefore, I am explaining this in a very every possible way. So write it down in your notes. The two things in your gut, these bacteria, the obligate, for your easy understanding, the obligate anaerobes are the good bacteria. Good bacteria. You can draw some emoji like this to understand properly. And in the second part, you can write the facultative anaerobes are the bad bacteria. You need to increase the composition, or you can say the concentration of the obligate anaerobes, and you need to cut off the or decrease the concentration of the facultative anaerobic. So for this, you need to always maintain the low oxygen concentration, and for this, even if little bit of oxygen is present, it will grow. So through this, you will be very easily, you can tell about what are the good bacteria in our gut, what are the bad bacteria in our gut. And the highlighted thing, the blue highlighted thing, you have to mention that how our body is fighting against different infection. So this is the thing. You also have to read about PPAR gamma. So PPAR gamma is a transcription factor which will bind to specific. What transcription factor will do? A transcription factor binds to a specific gene sequence, that is DNA sequence, and then it will start exerting its effect. So this is the thing. Now next part, this one. So this is a what to say, this is a comparative table, you can say, or the summarized mechanism through which our microbiota or the gut bacteria fight against different immunity and different infections. So how our microbiota provide immunity against different diseases. So the first and the foremost, we have studied this yesterday also, the T regulatory cell regulation, that our gut microbes promote the regulation of T regulatory cells. And how the T regulatory cells are produced? Through the short chain fatty acids, especially butyrate. Now, if you are observing or not, I don't know. I always try to provide questions for all of you people. Like, see, this question is, which of the following immune function is directly influenced by the gut microbiota? So I have included this because all these questions are a basis to completely understand the conceptual things of your topic. Whatever thing you are studying, food microbiology, gut microbiology, cancer biology, vaccine biology, or anything. So you should know about the basic conceptual thing in order to understand the advanced level things. So in this question, I have asked you that which of the following immune function is directly influenced by the gut microbiota? So here was the answer, that the regulation of T regulatory cells, which is under the control of the gut microbiota, they stimulate the growth of the T regulatory cell, which is ultimately involved in the inflammatory responses. So I have also included the explanation that especially butyrate, which even if you are asked, that examiner will ask that out of the following fatty acid, which is the most common for the activation or the stimulation of the T regulatory cell. So for this, butyrate is the most common one, which is responsible for stimulation of the T regulatory cell. So this is the question. Now here you can see this table that the T regulation is involved. SCFA will do what? The T cell regulation, and especially butyrate. So everything is interconnected. Even if you are missing today's class and you are directly jumping onto the next class, tomorrow's class, so you are missing so many things. So always try to have a coherence while studying. Even if you are missing for obvious reason, if you are having some class, your university exam or anything, I'm not saying about that those things, but try to finish the first day 1 class, then day 2, then day 3, day 4 likewise, not day first, then day 4, day 6. No, in this way you will not be able to understand the topic completely, because I am putting my all efforts that after the completion of all those things, you will be able to have an insight about what exactly is gut microbiota. You will be able to completely uh, uh, you will be able to know about each and every aspect of the gut microbiome. So I was explaining this T regulatory cell regulation. So how this is going on in our body. So our gut microbes will promote the regulation of the T regulatory cell, which is the third variant of the T cell in our body. So B and T cells are the part of adaptive immune system, not the innate immune system. Innate immune system is by birth, and adaptive is we achieve in our lifetime. So it will maintain immune tolerance, and it will reduce what? Autoimmunity. Autoimmune diseases will be reduced. Now the next mechanism is anti-inflammatory signaling. So anti-inflammatory signaling means what? Short chain fatty acid will do what? It will induce the anti-inflammatory pathways, and which SCFAs will maintain this? These three: butyrate, acetate, and propionate. So they will do what? They will bring down the concentration of interleukin 6, TNF alpha, and they will increase the concentration of interleukin, which is the anti-inflammatory cytokine. So you should know about what is going on in our body. So if any interleukin or if any chemokine is responsible for initiating any inflammatory responses in our body, then it will be automatically decreased by our immunity. If anything is supposed to cause infection in our body, what our body will do? Our body will decrease its concentration so that no inflammation will occur in our body. So it will bring down or decrease or cut off the concentration of interleukin 6, TNF alpha. TNF alpha is what? Tumor necrosis factor alpha, which is an again a pro-inflammatory cytokine that play a role in the immune system response to inflammation and infection. So it is a pro-inflammatory regulator. So we need to bring down the concentration of this TNF alpha. Now the third one is antigen presentation. So antigen presentation is what? Specialized gut cells present in the microbial antigen to immune cell. So the third mechanism is through the APCs. So APCs are antigen presenting cell, and they are the important part of our immunology. So they are the responsible cells which are required to present the antigens to the T cell, so that it will trigger the adaptive immune response. Like whenever your body is encountering any kind of pathogen, if a bacteria is trying to invade the body through the physical barrier, we have so many line of defense in the innate immunity. If this bacteria will try to invade the physical line of defense, and if this bacteria is capable of doing that, what our body will do? How our body will know that a bacteria has already entered in our body? So there is a mechanism which is known as the antigen presentation. So APCs are the cells which are responsible for this thing. They will make our T cell, that the major protector is T cells. T cells will do what? They will start the adaptive response. But for starting the adaptive response, they should know about the fact that our body is having bacteria and virus in our body already. The bacteria and viruses are entered in our body. So the APCs do what? The APCs are the, you can also say they are the antigen messengers. They are the antigen messengers. They will process the antigen and they will present it to the T cell that here is the antigen. Now I have done my part. I have sent this to you. I have let you know that our body has an encountered pathogen in our body. Now do your role. So this is the thing done by the APCs, antigen presenting cells. So there are so many APCs in our body like dendritic cell, macrophages, B cells. All these are APCs. They will hold what? They will hold the antigen, they will process the antigen and they will present to the T cell that we have encountered the pathogen, we have done our role, now do whatever role you are assigned. So T cell will do what? It will start its adaptive response to us, it whatever be so APCs are so many like dendritic cell, macrophages and B cell, and APCs are the antigen messenger that takes up the antigen from the pathogen and they process them and display them to the cell surface. And they do this with the help of MHCs. If you know about the MHCs, major histocompatibility complex. So this is an important part. If your body is not doing this part, this wala part, that is APCs, like if your body is having impaired APC cell, then how T cell will be knowing that somebody has already entered in your body? So this is the main part, or this is the crucial part in adaptive immune response, that the function of the APCs should be top notch so that it will always present the antigen to the T cell and then T cell will take care of everything. So some of the pathogen adapt this mechanism. They somehow escape from this APCs and they directly enter our body and they show so many inflammatory responses. So this is one mechanism. So they do this thing through the APCs. So what are the key microbial product here? M cells in the pairs patches. So M cells are the specialized epithelial cell found in the follicle associated epithelial of the pairs patches. That is the GALT. So we have already gut associated lymphoid tissue, which I have taught yesterday, that our body is having gut associated lymphoid tissue. So in that gut we have pairs patches. Pairs patches means we are having some specialized epithelial cell which are known as the M cell. These M cells are responsible for presenting those antigen to the T cell, to the responsible agent who will react towards it. So they will start activating the adaptive immune system and they will also maintain the tolerance level. Now the third mechanism is dysbiosis effect. Third mechanism or the strategy, dysbiosis effect. So whenever there is an imbalance in the gut microbiota, then the pathogenic bacteria will be in very high composition, high overgrowth. So it will increase the risk of inflammatory bowel disease, asthma, type 1 diabetes, and allergies. Now, there are some bacteria who can provide or modulate our immunity, like the Bacteroides fragilis, which will do what? It will modify the polysaccharide A, and then it will promote the T regulatory cell and it will suppress the inflammation. So this is one mechanism. And also there are so many mechanisms. Immune cell maturation, we are having so many armies or the fighters in our immune cell. There are so many macrophages. They will just do phagocytosis of the antigen and they will process them and they will eliminate them from our body. So they will not be able to cause any kind of disease in our body. So this table shows that how our body is protected from different kind of pathogens and how our body fight against any kind of pathogen and how our immunity is boosted through the gut microbiota. Now you can say that this is very important that you should maintain, and you have to maintain your gut micro biota composition very well, because all those things are important not only for your gut but for your overall health. We have studied up till now that how your gut microbiota is helping you to have a good gut brain axis, the MGB, a good immune axis, like how your body is protecting you to fight against different diseases, different infection and inflammatory responses. So uh, in order to remain healthy for longer, you need to maintain the good gut bacteria. If you are maintaining such condition, then your good bacteria will give you so many rewards. How they will reward you? They will reward you by protecting you from different diseases. They will protect you from different infections, from different inflammation, and also you will not have any kind of stress, anxiety, depression, or any other mental illness. So all these things are very important if you are having a good gut bacteria. So next part. This I was trying to explain. So the I have included this after whatever I have taught up till now, this wala part, because you have to explain this flow chart. So if your gut is having a good homeostatic condition, you can draw this emoji. Good gut bacteria means a happy you. So if your gut is in homeostatic condition, then it will do what? It will produce butyrate. Like this is the normal part. Up till now, you should be able to recall that what is the role of your gut microbiome. Your gut microbiota or your gut microorganism or gut bacteria will synthesize what? They will synthesize, they are able to synthesize short chain fatty acid like propionate, acetate, butyrate, but specifically for this role, like for the boosting our immunity, the major role is of butyrate. So our body will have, if your body is having a good gut composition, then obviously I have included this point that firmicutes and bifidobacteria will be in higher amount. So increased amount, you can see this arrow, increased amount of bifidobacteriaceae and firmicutes. So they will do what? They are the butyrate producing bacteria, so they will produce butyrate, simple. So they will produce short chain fatty acids, specifically butyrate. Now this butyrate will increase the integrity or the intactness of the intestinal barrier, and also it may enter the GPCR cycle, G protein coupled receptor pathway, and it will be involved in the decreasing the inflammation wala thing. Okay. Now this SCFAs will do what? I have already explained the PPAR gamma. So this PPAR gamma will do what? It will decrease the nitric oxide synthase level, and also it will decrease the nitric oxide, and then it will increase the beta oxidation to meet the energy expenditure, and a low oxygen condition is required, which will support the growth of bifidobacteria and firmicutes, which are the obligate anaerobes. If I have not included this thing previously, and directly if I will produce this wala part, so you will not be able to understand it. So therefore I have explained this in a very detail, that PPR gamma has so many role here. It is the responsible regulator or the transcription factor for so many things. So here you can now explain this very easily. So these are the cells of your gut. These are the IEC, intestinal epithelial cells. Everything is going on inside the intestinal epithelial cell. So if your gut is having a good homeostatic condition, obligate anaerobe concentration will be increased, specifically bifidobacteria and firmicutes. So they will produce butyrate, and then this butyrate will increase the concentration of PPAR gamma, and it will increase the mitochondrial oxidation, and it will decrease the epithelial oxygenation. And SCFAs also directly bind to the epithelial and immune cells G protein coupled receptor that reduces the gut inflammation, such as GPR41, GPR43, and GPR109A. So you should know about the GPCR cycle, the serpentine receptor, 7 transmembrane receptors, G protein coupled receptor. So through these pathway, like it can also enter the GPCR cycle pathway, and also the activation of the PPAR gamma. So this is the condition when your body is having a good gut microbiome composition. So you should know about all these things about a good homeostatic condition. Now the next part, like this is the condition when your gut is healthy, but we also have somebody whose gut is not healthy. So the second condition, if your gut is not healthy, then what is going on in your body? So instead of the good bacteria, bad bacteria will increase. What kind of bad bacteria can you name? These are the obligate anaerobe. So what? Facultative anaerobe. Facultative anaerobe concentration will be increased, and they will directly involve in the inflammation. They will increase the inflammation, and also they will not will the synthesis of butyrate, and if butyrate is not synthesizing, PPAR gamma will also be not synthesized, and then NOS, nitric oxide level will be increased. So this will inhibit what? This sign means what? Inhibit. This sign means inhibit. So this will inhibit beta oxidation, and then anaerobic glycolysis will start, and your glucose will be converted into lactate. That is known as Warburg effect. Warburg effect, and then Warburg effect will increase what? Your gut lumen oxygen concentration will be increased. Then it will support the concentration of the facultative anaerobes. So now see, here low oxygen condition is there because beta oxidation always takes up all the oxygen, and in here if it is anaerobic glycolysis, then oxygen will be available because oxygen is not utilized in any of the metabolic pathway. So therefore first thing is, if your gut dysbiosis is there, you will increase the concentration of all the unhealthy bacteria, or you can say the facultative anaerobic. So enterobacteriaceae, pathogenic bacteria concentration will be increased. Then it will decrease the concentration of the SEFAs, that is butyrate. Now this will inhibit the PPAR gamma. In the good gut bacteria, it will stimulate the PPAR gamma, and the stimulation of PPAR gamma will do what? It will increase the beta oxidation. It will just cut off the oxygen level. So these things are going on if your body is not having a good gut composition. So you can read here that the interaction between the microbiota derived metabolism and the gut epithelium. If your gut dysbiosis will occur, enterobacteriaceae will increase, and it will just increase the inflammation in your body, and the integrity of the intestinal barrier will decrease. Now what condition we can say this? Leaky gut. Leaky gut will do what? It will just throw all the inflammatory molecules in your bloodstream, and your blood will be full of all the inflammatory mediators, and then this is a very bad thing in your blood. Our blood should be purified. It should have always a good composition of all the formed element. Formed element means RBC, WBCs, and all the other thing, hemoglobin 2, platelets 2. So if you are having plasma, good amount of plasma, 45 plus 55% composition is there in our blood plasma and all the formed element. So if your body is having high amount of inflammation, just because if you are not having, you have not maintained a good gut microbiome composition, then all these things will be happening in your body. So this is the thing going on in your body in the context of immunological thing, immunity and inflammation. So I hope I have explained this. I have explained this clearly. So try to have a clear cut notes in your notebook, because all these things are very important and are very specific. You cannot say our gut microbiota protects our body against different pathogen, and it boosts our immunity without having a solid mechanism, without having a specific mechanism or pathway. You should know about the exact pathway, exact molecules, and how they do it. You cannot say superficially that it. Superficially, we all know that our good bacteria will do good thing in our body. They will increase our immunity. They will prevent any kind of infection. But how they do it, you should know about it. So make pointers out of all the things I have taught to you. Try to make a crisp notes, no lengthy notes. Always try to make notes as per what your examiner asks in the CSIR-B section, because in CSIR-B section we are always asked a very specific question, to the point question. So try to make notes like that way only, because nobody likes to read a long paragraph. So try to have, you can draw this flowchart directly in your copy. So all these things are from different papers, because no book is available with having information about all the gut microbiota thing. They are having a chunks, they are having some sort of information about how they do it. Some pathways are known, but majorly we have to refer different literature to have insights about the exact pathway which is involved in controlling or influencing our different axes. So this is all for today. We will study this part tomorrow. Like this is the actually compiled table. Like factor for dysbiosis are antibiotic treatment, if you're not taking good diet, physical damage to the mucosa. So I will explain this tomorrow. This is nothing but the same thing, this diagram and this one. Like if you are having a gut homeostatic condition and if you are having a gut dysbiosis, so A plus B. So I've compiled this and what it can happen. So here I was talking about this wala part, that your body, if you are having a leaky gut, all these inflammatory molecules will be in your bloodstream. So your blood will be unhealthy. So we will talk about this tomorrow. So let me stop the recording. So we will